Okay, so with that I finish the alginate part, and I will just sum up what we talked about. First, we talked about this very important transition from M to G, leading to a conformational change from 4C1 to 1C4. We talked about the G blocks and the dimerization with calcium leading to gels. We talked about the elasticity and especially how it depended on the the G block length, how to make beads and capsules. We talked about homogeneity, swelling and stability. I mentioned the GDL method for homogeneous gels. And I used an example, the encapsulation of Langerhans islet for diabetes, which led to immune stimulation and protection, and finally how to make alginate. So I think that's it. So if you have questions about alginate, you can come to me afterwards if you like. So I plan to use the last 20 minutes or so on something different. Chitosands. So chitosands are also marine polysaccharide, but not from algae, but from shrimp. There are many types of shrimp. In Norway, we harvest a small shrimp called Pendulus borealis. It's about this size. It's caught outside Norway, and it's cooked, so it becomes red, and we peel and eat the inside. It's very delicious, especially when it's fresh. So you can, students in Trondheim sometimes go and buy half a kilo of shrimp and some bottles of beer and go and have a picnic. The shell of the shrimp, but not only shrimp, also in insects have chitosan, and even squid has chitosan in some cases. So in the shrimp industry, the shell is collected, and it goes through a process to remove proteins and calcium carbonate. You know that the shell is rich in calcium carbonate and special proteins. So after removing those, in a very tough process involving both acid and alkali, we obtain almost pure chitin. Another source of chitin is some fungus that you can grow in the lab, but they produce very small amount of chitin. But it may be very important in biotechnology. The chemical structure of chitin is well known to most people, to most life scientists. It is very similar to cellulose. It has only one sugar, and what makes it different from glucose is this part. If this had been OH, it would be glucose, and it would be cellulose. But this is another sugar. It's called N-acetyl-glucosamine. So there is a nitrogen on position 2 linked to an acetyl group. Biochemists often abbreviate this by GLUCNAC, especially in glycobiology textbooks. Chitosan people call it A for the acetyl group. So the chitin undergoes alkaline hydrolysis under very strong conditions to obtain chitosands. And chitosan is chitin where we have removed most of the acetyl group, obtaining a free amino group like this. And typically such a process removes about 80 to 90% of the acetyl groups. So if you buy chitosan from supplier, there's typically 10 to 20% left of this. The interesting thing is this amino function, because that is a base. So it will attract proton and it becomes positively charged. The new sugar is just called glucosamine, GLCN. We call it often D, not because of this, but because it's deacetylated. Yeah. So chitosan will be a polycation because of this equilibrium. The pKa value is about 6.5. That is a little below physiological pH. So if you want to use chitosan in with human cells at pH 7.4, we have about 10% charges and 90% no charge. So it's weakly positively charged. But because of this, it binds very well to cell surfaces, for instance, which have negative charge. So in the chitosan field, we also have a composition and sequence problem, or a sequence situation. We can define fractions just as we did for alginate. But in this case, we have Fa and Fd instead of Fg and Fm. So what about the blocks? It turns out that the deacetylation process is random. Very different from how alginate is made. Therefore, what you have left is statistically distributed. And because of this, we can predict directly that, for instance, the AA dyad frequency is just Fa squared. And if you want to have this tetrade, it's simply 3 A's and a D, so it's a frequency of A to the power 3 times D Fd, and so on. So this is a very different situation from alginate and makes things a little easier for us. So commercial chitosans will have typically Fa for from 0.1 to 0.2, meaning from 90 to 80% deacetylation. And molecular weight is in the range 2 to 400,000 typically. Chitosans are used in many different ways. I know Japan has been one of the leading countries in using chitosan for water purification. Some years ago, it was a very popular slimming agent, especially in the United States. There is a patent saying that it lowers blood cholesterol. My colleague says it's probably true. But I don't know. It has been used in cosmetics, in biomedicine, especially in nasal drug delivery and gene therapy. I will talk about that in a minute. And finally, you find many research articles claiming that chitosans are antibacterial. I put a question mark because I personally think that the literature is too conflicting to conclude.